Hello and welcome to Invertebrate Paleontology and Paleobotany. This is lecture 3, where we'll try to answer the question, what is ontogenetic variation? So to begin our discussion on ontogeny, I want you to look at this interesting bug here. This is a bug that's extremely common throughout the world. You should be able to recognize it, and some of you might recognize what this bug is. This is a juvenile form, a larval stage of a bug that you're probably a little bit more familiar with, and that is the ladybug. So what ontogeny is, and what ontogenetic variation has to do with, is how organisms change through time as they grow. So growth stages in organisms may be very different, and if you're trying to recognize fossil specimens and you only had fossils of ladybugs, if you saw their juvenile larval stage, you'd probably think that they're a completely different type of creature and not the same species. And we're going to encounter this problem throughout the fossil record, is trying to untangle the ontogenetic series of fossils and how they change their shape and their features as they get older. So ontogenetic variation deals with the growth stages and how we might be able to recognize those growth stages in the fossil record. So it doesn't necessarily need to be as extreme as we see with the insects like ladybugs. This is a growth series showing humans, our own species, basically using the same proportions to show how different parts of our bodies change as we age and get older and we grow. So babies have huge heads than, say, an adult. And so you can see these proportions change through time, and this is what we're going to talk about, is ontogeny. So imagine that you had a bunch of series of these fossils. You would probably think humans were a bunch of different species, with the babies being completely different species than the adults. With a lot of specimens and fossils, they actually go through growth stages like we see with ladybugs. Trilobites are a good example of that. So trilobites are members of the arthropods, and arthropods have exoskeletons. So instead of internal skeletons and growing kind of like mammals do, like we do, they have exoskeleton that shed at certain stages. And so each time they shed, they can change their shape and morphology. And so in the fossil record, you get these sort of stages of trilobites, juveniles, and then you eventually get adult styles of trilobites. So this makes it really tricky when you're trying to identify specimens of trilobites. So let's talk a little bit about the types of growth that we see, and then we can use that to try to figure and untangle these things in the fossil record. So one of the types of ways organisms can grow is simply by the accretion of existing parts. A lot of the bivalves and brachiopods that we'll talk about this semester use this type of growth. Basically what they do is their shells or exoskeletons will grow by just adding a little bit more to the skeleton. And so over time they become bigger by just adding more of that skeleton. So think of how a shell grows. This is a scallop, and we see that there's a growth each year, sort of annual, sometimes even monthly growth stages in these shells, and that basically keeps adding on that existing exoskeleton. Sometimes it can be a little bit more complicated. Instead of just adding on to the end, here is a bivalve showing how bivalves can actually add a little bit to the outer external part and thicken up the shell. So the growth lines here are actually kind of curved, and this is something important that you need to consider if you're going to use some geochemical analysis on those growth rings of some of these shells like brachiopods and bivalves, is to know exactly what those growth rings represent, because they kind of curve over. So in bivalves they kind of add a little bit, thicken up the shell. By thickening up the shell as they get bigger too, that helps to protect the inside of the organism from any predators. Another type of growth pattern that we see in the fossil record and with living creatures is just simply the addition of new parts. And probably the best example of that is with a group of echinoderms called crinoids. We'll talk more about crinoids later this semester, but these are organisms that live on stalks that live on the bottoms of the ocean in marine areas have a very extensive fossil record. And the way they actually grow is by adding segments to their stalks. So this is showing how a crinoid sort of grows by adding these little discs to the stem of the crinoid right below sort of the flowering head, which is called the calyx. And it's in that portion that they can grow, so they get taller and longer the longer time that they're around. What's really kind of fascinating is when you have a large population of fossils that you can sample, you can categorize them as being young or old individuals. And with crinoids, there's a very unusual and interesting ontological stages. As these organisms get older, they change. And so here's an example of young crinoids of the same species as well as old ones. And you'll notice that as they age and get older, they tend to thin out their calyxes in terms of the finger projections that are on top of there, as well as thinning out, but making it much longer, the stalks that they build on. So the width doesn't change, it's basically adding those segments as they grow, which is a really interesting and unusual way in which organisms can grow and change through time. So this is really important to recognize, because each one of these growth stages might be recognized as a separate species or a separate organism. The other way to grow is through molting, and this we see in insects, in arthropods, in many types of invertebrates, and we'll talk a lot about it with trilobites. So trilobites have growth stages where they molt. They molt their exoskeleton, and this is true of all organisms that have exoskeletons, is that they need to grow, and you have an external exoskeleton, you need to grow a larger skeleton underneath that, and you shed that. And in fact, one of the reasons I think trilobites are very common in the fossil record is a lot of the fossils actually represent the molted off portions of their skeleton. So every time they go through a stage of molting, they lose that skeleton, and sometimes you find that molt in the fossil record rather than the actual organism that lived. So it's kind of fascinating. So one of the things that happens with each molt is that the shape and morphology of those trilobites changes through each molt. And so one of the things that paleontologists try to do is try to look at each of the molt stages of trilobites and recognize whether they're the same species or a different species. So whenever they get a large population of trilobites, try to recognize where these molt stages might appear. And here's some examples of some different types of genera of trilobites and different molt stages that they exhibit throughout their growth patterns. So one way you can actually do that is by simply doing lots of measurements of fossils. So here's an example of the total head length of the and width of each of these trilobites that are found in a large population of fossil trilobites. So measuring those, and one of the things you'll notice when you look at this graph is that they sort of cluster in little patterns, and each one of those represents a molt stage. So when they're young, then they molt and they jump up to another higher category. And so this is one way you could actually look at a population and categorize each of the individual fossil specimens you have to each one of these growth stages, which represent different molt patterns. So one of the things, if you go into trilobites and research on trilobites, is basically recognizing each one of these molts that happen for each of the species, and that way you can sort of categorize things. And it gets kind of complicated because each one of these molt stages can represent a significant shift in the shape and morphology of the trilobite, although they overall are getting larger through each molt. One of the things we can also do is look at growth curves. So growth curves you're probably familiar with when you were a child and went to the doctor, and they would measure your weight and height, and they'd plot your growth onto a growth curve. And you probably remember that everybody had a different projection in terms of their growth. So sometimes you're below the growth curve, sometimes you're above the growth curve, sometimes you're in the 20th percentile, sometimes you're in the 70th percentile. And so that just basically meant where your particular body exhibited on that growth curve. And so everybody has different growth projections, and these are two graphs that show that. So this is looking at length and width with many different individuals of different ages plotted on here, and you get this kind of growth curve. Everybody gets bigger over time, but we do it in different ways. Sometimes we jump on that growth curve, sometimes we don't. Sometimes we may not be eating as much, and sometimes we hit a growth spurt and start growing very quickly. So we can actually kind of look at growth curves and attempt to think about that when we're actually describing and working with fossils. So one of the interesting things is that there's a lot of different types of organisms in which as they grow they kind of keep the same shape. And one of the great examples of that is in cephalopods, in a group called the ammonites and the nautilus. So this is a nautilus shell. We'll talk more about this when we talk about mollusca. But this, what happens is as the shell grows, it adds an additional chamber. So the shape doesn't change at all. In fact, the shape is really important for these because if they do change, it makes it more difficult for those organisms to survive because they use their shell as a buoyancy when they swim in the ocean. So if they change their shape, they'll have problems functioning. So it's got to be functional as they grow. So what's interesting though is as those revolutions are added, so each one of those chambers represents a growth stage of this shell, each of those revolutions will add larger and larger chamber size because it has to keep that same shape. So what this graph is actually showing is the radius of the shell versus the angular position of the aperture. So this is sort of an exponential increase. So each of those apertures have to get much bigger through each revolution as it goes if it wants to maintain the same dimensions, the same shape. And so this is one of the classic mathematical examples of increasing growth, the golden ratio in terms of the way in which the shell grows through time. This is one of the constraints of growth, is that as you grow, you have to basically reach these maximum sizes just because you have to produce the next growth stage, you have to grow even bigger. And so you can see in this graph here that as each one of these growth stages and it gets bigger and bigger, the aperture, the size of this opening has to get larger and larger as it goes, and the angle here. So you can see that with each growth stage they have to add a larger chamber in here. So some of you might be familiar with Fibonacci numbers. This is a sequence of numbers that were derived about the year 1200 AD by a mathematician named Fibonacci. Basically what it is that he realized that he could take proportions and increase them. So the series begins like this. You start with 0, you then and 1, you add 0 and 1, you get 1, you add 1 and 1, you get 2, you add 1 and 2, you get 3, you add 2 and 3, you get 5, you add 3 and 5, you get 8, you add 5 and 8, you get 13, you add 8 and 13, you get 21, and so on. And this was shown as sort of like a way to increase exponentially a series. And what's really fascinating is, like, for example, here is a diagram of showing how he actually put together it. So he basically is counting each of those squares to show how you can increase exponentially the size of something. But at the same time, what's really fascinating is that it keeps the same shape. So each of those shapes are the same as you add each of those components. And you see this a lot in nature when we look at how things grow over time. Because they want to keep the same shape, they're going to follow those unique sort of series of numbers in their exponential growth as the creature grows. One of the probably most fascinating insights into how organisms can change their proportions was developed by Darcy Wentworth Thompson, who is a scientist, wrote a famous book on growth and form. And he developed a technique called coordinate transformation. This is a idea that now is basically in every single textbook in biology, and particularly in embryology. That is the study of how organisms basically develop from a very early stage. So one of the things that he discovered was that you can actually change the sizes of certain coordinates within a organism, and that can lead to radically different types of organisms. So in the examples in his book on growth and form, you can see some examples of fish. Both of these fish that are depicted are actually species. But what he showed is that in changing the the pattern of the growth of the proportions of each of those creatures, you actually could easily transition from one species into another species by simply making simple changes in the amount of growth in each of those coordinates that he did. And he did these with really wonderful illustrations of showing how this transformation occurs. One of his illustrations that he has in this book is with the human skull and comparing that with a chimpanzee skull. So here we have the chimpanzee skull and we have a human skull, and one of the things that you can quickly recognize is how bigger and larger the size of the cranium has become in humans versus chimpanzees, and how small the basocranium, the base of the skull, has reduced the amount of growth in humans, and this has brought the face down. You also see that the jaws have gotten much smaller compared to a chimpanzee. And so this is one of the early ways in which we could actually look at the growth of certain portions of an organism and how those changes could actually give us insight into how we evolve from different types of groups. So wonderful, wonderful analysis. Now we can use computers and using coordinate analysis and do some really fancy things. But this is where it got its start, almost 100 years ago. So let's talk a little bit about growth rates, because growth rates can change. So you think about when you're a child and you're growing up, or I have 2 daughters and they are actually growing, and it seems like this summer they both just shot up in height and just put on a couple inches over just a short period of time of a couple of months. So growth rates can vary quite a bit. And here's an illustration of what we see when we look at growth rates. So the first graph that we see down here, here we have the shape of something. This is a good height or length of your finger or something like that over time. And here, we have the babies start out. We have a period of adolescence where there's a lot of growth, and then growth starts to slow down later on in life. Now, not all organisms have to exhibit that pattern. Some organisms can continue to grow throughout their lives, like the ammonites. So as they keep growing, they'll keep getting bigger and bigger at the same dimensions, and it keeps getting larger. But a lot of organisms will actually slow down or stop growing as they reach a certain size. And some organisms will actually try to resorb or make some major changes in their bodies as they reach maturity. One of the things we can do is apply some calculus with this. So this is taking the derivative of that. So this is the sort of acceleration of growth. And what this is showing us is what time periods in an organism's life in which most of the growth is done. And you can see that it's mostly in the adolescence period, so that the height, that peak in that curve, represents the greatest amount of acceleration of growth, which occurs during adolescence in this case. And the other thing you can do is take the second derivative, and this is actually showing where you actually have a sort of a negative pattern that you see later in life, and that most of that growth occurs in the very first part of the organism's life. So you can do some really fancy mathematics that you can apply to these things using calculus, for those of you who know calculus. All right, so there's 2 types or styles of growth that we see in ontogeny. The first is what's called isomeric, and that is where the ratio between the sizes of 2 parts of an organism does not change during ontogeny. The other type is anaisomeric, and anaisomeric is where the ratio does change during ontogeny. So let me give you an example of that. So isomeric growth, if an organism exhibited isomeric growth, it would mean that the babies would look identical in proportions to the adult. So this is like if you were to slide a file size up and down in like a Photoshop or something like that and change the size of something. You're going from something small to adult, but you're not changing the proportions. So it's just, you're just sort of scaling up. So it's like you're zooming in and zooming out on these organisms. Isomeric growth is very rare, and oftentimes it's functionally constrained when we see isomeric growth. Anisomeric growth is where the proportions change. We see this with humans. So with humans, anisomeric growth means the proportions change relative. So we have much bigger heads in proportion to our bodies as an infant than we do as an adults. So I don't know if you guys are familiar with the artwork of Nina Levy. She does a lot of like crazy weird art, weird stuff, and one of the things that she plays with is ontogeny. So she takes young kids, these are her kids, and sort of applies sort of grown-up parts to them and makes it look absurd, or she takes adults and applies sort of baby features onto adults, and it makes these people look really creepy, even though the kid is not really creepy, because we're used to seeing these big heads in children, but we're not used to seeing them in adults. And so she plays a lot with this. She also does these big, huge, giant statues of babies. And the reason that it's so disturbing, these images, is because she's playing with ontogeny, and she's twisting around ontogeny, and she's trying to make these humans become isomeric in their growth and in the same proportions. And it just is absurd, and it makes you kind of have visceral feeling to it. So definitely check out some of her artwork. Very cool stuff. So where this might be of importance in paleontology is in trying to determine whether these organisms are juveniles or adults. And a really good example of that was a discovery that was made about 15 years ago in the island of Flores in Indonesia. And it was a discovery made in a cave. The fossils dated to about 18,000 years old. And they were remarkable skulls that were sort of like mini versions of modern humans. Here's an example of the skull that was discovered with a modern human for scale. And people started to debate whether these skeletons were a separate species of sort of dwarfed human, or whether they were some sort of disease that occurred, or just juveniles that they had discovered. And so you encounter these types of problems a lot in paleontology. So one of the things they had to determine was whether these fossils represented juvenile forms of humans, or maybe some sort of diseased or smaller version of humans, or whether they were completely separate species. This is Bill Jungers, who's a professor at Stony Brook, who does a lot of work on allometry in early humans. And he's a big guy, and he's standing next to a model of one of these creatures that was discovered in Florence. The people who described that it's a separate species is known as Homo Florencia. Below I have a skull of a juvenile modern human skull, showing that that there are some differences. The biggest difference is in the eruption pattern of the teeth. So these skulls had a eruption pattern that is very similar to what we see in adult humans. So one of the things that scientists started to do was to try to look at these skeletons to see if they actually represented in that growth curve of humans. They were very lucky and very fortunate to actually have a fairly pretty complete skeleton that had a radius and a femur. So this graph here that I'm showing on this side here shows the radius length and femur length, and they've plotted in here humans, which is this graph here in red, showing that trajectory. They also put in gorillas and chimps, so chimpanzees. So each of these curves kind of represents a trajectory, a growth trajectory. And so we would expect if these are sort of juvenile modern humans, they would fall within that line. The interesting thing is that skeleton there plots out where it's kind of shaded in yellow there, so indicating that it's not on the same growth pattern as modern humans. So what this is showing, that as the radius gets larger and the femur gets larger, these proportions are played out among each other. So looking at two different types of measurements and then scaling these and seeing how they change through time. Both those measurements are going to increase as the organism gets older, and then projecting those on can help you sort of unravel whether you're dealing with a juvenile or an adult in these cases. So if we look at all of life out there, when we look at these growth curves to see how organisms change as they get older, isomeric growth is very rare, and isomeric growth is much more common. And so here's kind of some graphs to show why that might be. So isomeric growth is basically the proportions are staying exactly the same. So sometimes you can have changes. So isomeric growth would be this first graph here, up here, where the proportions of y and x remain the same. So these are mini versions of the version up here. But you can kind of play around a little bit with the growth. So in each of these cases here, where as it grows, the width is growing faster than the height, and so that leads to longer and longer rectangles as it gets larger. And this might have to do with functional constraints, and I'll talk more about that in a second. But you can also see changes that happen. Very common, you'll see growth trajectories that are much more common, like C, and that is where there's a change. So the slope will change through time. But sometimes you can have a combination of these things where the slope is not 1 and totally equal. So you can have organisms getting wider because as they get bigger, they get heavier and they need to support that additional weight. So a lot of times it's functional constraints that make these organisms not behave like an isomeric growth pattern. So these are things to be aware of when we look at ontogeny. So I mentioned the word allometry. Allometry is basically the study of looking at lots of measurements of different organisms of different sizes. So allometry also includes looking at closely related organisms that have different sizes and the constraints that those sizes have to determine like why they might be getting wider as they get bigger to support weight, or has some sort of functional reason for that change in their proportions. We're going to be reading a paper by Stephen J. Gould, classic paper looking at Irish elk. These are an extinct group of elk that lived not only in Ireland but across Europe that had these huge antlers, and looking at how the growth of those antlers could be tracked through allometry, and why they might have had these giant huge antlers and which individuals had those giant huge antlers. So Stephen J. Gould traveled all around Europe looking at skeletons, knocking on people's doors that had skeletons and measuring them, and it's a wonderful, wonderful paper looking at ontogeny, and the growth series is a very classic paper, so we'll be looking at that, and it's a really cool group, even though they're not invertebrates. So one of the things I want to talk a little bit about is an analysis you might encounter when you're looking at ontological variation, and that is an analysis called principal component analysis. Oftentimes it's abbreviated PCA, and you'll probably encounter this in a lot of scientific literature when you're looking at morphometrics and looking at sort of quantifying and measuring specimens. So this is an example that comes out of a paper on Homo Florensis that I mentioned a little bit earlier, looking at a bunch of measurements of the skulls. So what principal component analysis does is it basically, you take lots and lots of measurements of fossil specimens. You may be using one of those microscribes or lasers or something like that. You have tons of numbers. You plug those numbers in and you ask, where in our sample do we see the greatest amount of variation? And so it'll spit out some sort of parameter. And that will be the principal component, the first principal component. Then you ask, all right, what's the next sort of feature that shows the next greatest amount of variation? And it spits out another sort of feature, and that would be the second principal component. So you take those two, sometimes you can take up to 4, and then you map those, you graph those. So these are comparisons in sort of a made-up space. And it sounds kind of confusing, and oftentimes when you see these, what you're looking for is how separate each of those samples are. So each of those fossils, how separate can you separate those things out? So in this paper, they're looking at whether Homo florensis was a separate species, then looking at humans, looking at little humans like juveniles, and looking at humans that have some sort of problem in their skulls, and trying to see if you could tease those things out. For example, in this analysis, you're like, what measurements are there? And they give the actual measurements. So these are the measurements that they used. This is in the skull. So they're looking at each one of these. So A is the principal component, which basically, looking at it, it's kind of how squished the occipital, the back of the brain case is. The second one that they saw the most amount of variation is kind of how squished the skull is. Based on those measurements. So that's all their points. So they plug those into a computer and analyze it. So when you look at a principal component analysis, you're basically looking at how separate are each of those populations, and then you can go back and usually they will define in the paper what those principal components are. And oftentimes they're difficult, so in this case they were using a microscribe, and so these are certain key points on the skull and the differences. So now let's talk a little bit about why form changes with growth. Why do we see that young individuals are completely different than adult individuals? And a lot of it has to do with a change in the mode of life. And throughout this semester, we're going to be highlighting a lot of groups that undergo a huge amount of change as they mature, and they go through a very complicated life cycle. So sometimes you can even have organisms that are the same species that will have different life cycles in which the smallest version is completely different than the larger version, or different stages in their life. So one of the things that happens as an organism gets bigger is that there's a bunch of functional constraints, mathematical constraints on how that organism has to function. So in this graph here we see a variety of different organisms plotted by body weight and volume distribution. So what happens is that as they get bigger and you add more body weight, the volume also has to increase to accommodate that body weight. So all sorts of morphological things can happen. So for example, bones have to be bigger and thicker to support that addition of weight. A lot of it has to do with a mathematical property that happens in terms of surface area to volume ratios. So let me explain that a little bit better. So the surface to volume ratios change. So if you have little organisms and they all stay little, the ratio stays the same if they're broken into multiple units. But as they get bigger or expand, they may not necessarily be able to support that increased weight or that increased volume that they have to contend with. And so this is one of the reasons why some of these changes can happen as an organism gets larger, is to deal with the functional constraints of that change in surface to volume ratio. So we'll talk a little bit about colonial organisms in which ontogeny can also have an effect, but the ontogeny may not necessarily be at the individual level, but at the colony. So we'll be looking at fossil corals throughout this semester. So fossil corals are kind of behave like a city in terms of each one of the polyps grows in each one of these cavities as an individual, but they live in a colony. And so as the colony gets larger, it's going to have functional constraints in how large it can get. So thank you for watching, and if you're interested in taking a course at Utah State University and learning more about paleontology, check out our website at geology.usu.edu. And if you're interested in some of the research that I do and a little bit more about who I am, check out my webpage at benjamin-berger.org. Thank you for watching.